a discussion about the database landscape, specifically what we're doing within the clinical research realm, and what the deal is with all these consent forms. When he's done speaking, we're going to be joined by Dr. Eric Marsh, who will talk a bit about biomarkers and how they fit into the clinical research landscape. With that, I'm going to let Dr. Demarest start his talk.
All right. Thank you, Heidi, for the introduction. Thanks for the invitation to chat about this topic. I always start off with some disclosures, and I've worked with a couple of companies on unrelated matters. So what I want to get across today is what types of clinical research we do and why those different types of clinical research are important to the community, really, to all of you. I'm going to give you an update about the ICCRN. I'll define that for you, don't worry. I know it's been thrown around a little bit by Helen in the last talk. An update on the severity assessment that has also been discussed a little bit, as well as some of our other outcome measures that we're developing along with Helen Leonard and Jenny Downs. And what do we need from all of you, the families of patients with CDKL5 deficiency disorder? Finally, I want to give you some sense of what we're trying to do to streamline all of these complicated databases and networks so that information can be used as efficiently as possible, but also safely. So I'll give you a sense of that. And then I'm going to turn it over to Eric to talk about biomarkers, and I think he and I will take questions together at the end. So types of clinical research. Within clinical research, you can really think about two big categories of research. There are interventional trials and there are observational trials. Interventional trials are what we think about as drug trials. This is where we actually do something we otherwise wouldn't do to a patient to see if that helps, if it makes something better. So the trials that have been done mostly so far in CDKL5 deficiency disorder have been epilepsy trials. And we could think about the Ovid trial or the Demarest trial. There's also the potential we are looking forward to of other types of trials that might be trying to address the underlying problem with CDKL5 deficiency disorder. Those would be things like gene therapy trials. There's things called ASOs or enzyme replacements. And there's a number of different strategies that different groups are working on to try to bring those therapies forward to a point where they'd be ready for a clinical trial. So that's one type of clinical research. And it's actually not my primary focus of my talk today. Those clinical trials are set up by somebody who has a drug and it's run according to the way that they think they need to run that trial. And I'll talk a little bit more about that. But there's also these observational trials. And that's most of what you've heard about today. The ICDD, the CDKL5 registry are observational studies where we are collecting data without any particular intervention. We're not manipulating what's happening to patients or what type of care they're receiving. We're just trying to capture, observe that what is happening to patients and see how that changes over time so we can learn more about CDKL5 deficiency disorder. So some of the ways that that might happen would be patients getting standard treatment, researchers are collecting the data, and we want to answer certain questions about the phenotype. I'll explain what that word means, the natural history, biomarkers, and outcome measures. And I'm going to go through all of those things. And these studies really are necessary in order to be ready for drug trials, particularly when we're thinking about these drug trials that are maybe trying to fix the underlying condition in CDD. So what is the goal of an observational research? I mentioned phenotyping, natural history studies, and outcome measures. These are three big goals. They're not the only goals, but they're the big ones. Phenotyping is describing common and uncommon features in CDD. What happens to patients? What kind of problems and symptoms do they have? Natural history study is looking at how do those symptoms or features change over time? How common are they? How severe are they? And how does a feature predict something that we care about in terms of outcome, like development or whether epilepsy is going to persist or how severe it might be. And finally, we like with natural history studies to be able to also include a biomarker component. Again, I'm going to mention that that's important in terms of understanding how certain things relate to each other and making good predictions, but I'm going to leave most of that discussion to Dr. Marsh. Finally, outcome measures is how do we accurately, reliably, and quantifiably capture CDD? And that's the really challenging part. We could write a book about each patient, and it would be very accurate and describe all the things that are going on with them. But how do we compare one book to another book? How do we compare your book to your future book after some sort of intervention? And so we have to start to move towards quantifiable ways of being able to capture this without losing too much information. So when we think about phenotypes for CDD, we could think about visual impairment, development, or epilepsy. And those are key features of CDD for sure. But CDD is actually a lot more like this. It's a huge number of different symptoms that patients and families have to deal with. And it's challenging then as a researcher to figure out how do you make sense of all that and organize it in a way that you can understand. And that's what we're trying to do with a good natural history study. So it's not good enough for us to just say that some patients with CDD have problems with development and epilepsy and sleep and GI and CVI and respiratory and behavior problems and so on and so forth. We really need to say, how often do patients have those symptoms? How severe are they? How do they change over time? What predicts a change? How does one symptom relate to another symptom? And how can we best treat these symptoms? And those are all questions we try to answer with a natural history study that goes one step beyond just phenotyping, which is basically describing what kinds of challenges they have. So the question then becomes, can we take this and turn it into this? Can we organize that chaotic group of information, all the different things that are happening with a patient, into something that we can make sense of and track over time? So why does this matter? Why do we need observational research? With rare conditions, it's actually very helpful to clinicians who are less familiar with a condition just to have good descriptions in the medical literature about what's going on with patients. That helps them to be able to treat patients better. It also helps us to prioritize what types of treatments might be most effective or important. And it helps us avoid unnecessary testing for patients when we have a better sense of what kinds of things we should be ready for and watching for. Treatment, how do we know if we're making a difference on something if we don't know what's typical in patients with CDD? So a natural history study tells us what is typical for patients with CDD and what's the range of typical. And if we don't have a good answer for that, it makes it hard for us to have a really good interventional trial and change the course of that disease. This helps us prioritize research, which symptoms have the biggest impact, which ones are most important to families. It helps us identify clinical features that may be useful to track for a clinical trial. So what types of clinical features are things that we can measure appropriately, that have a big impact, that are going to be important for us to follow? And that's a process of developing what we call clinical trial readiness. And that's a buzz phrase that's been thrown around some at various meetings. And I want to provide a little bit more detail about what that is and what it might look like. So what does it mean to be clinical trial ready? Really what we're trying to get at here is, if we do a drug trial, will we get the right answer? Another way of saying that is, if the drug works, will the results of the trial show that the drug works? That's critical. If we do a trial and a drug really does work and it's making patients better, but we can't show it, then that drug may not get approved by regulatory agencies like the FDA. And then it won't be available to future patients. And that to me, is the most heartbreaking possible scenario. So what does it take to get there? Why would we not get the right answer? Well, if we don't measure change correctly, that has to do with outcome measures, then we may not get that answer. What if we don't watch the change long enough? What if we don't have enough patience? Those are all aspects of a trial that can be informed by natural history studies and by clinical trial readiness studies. So more about outcome measures. Why are they important? What do they do? They're how we measure if a treatment is working. And they have to fit the symptoms that we're trying to change and capture the range of symptoms that patients with CDD have. So there's a couple of ways that this may be problematic, and I'm going to try to demonstrate some of those in a sort of theoretical way. So there's effects called the floor and ceiling effect. There's appropriate discrimination. This has to do with how far apart the intervals are on a measure. And the distribution. What's the spread like? Does it capture the whole population? So ceiling effect. Whatever measure this might be, if it's measured from 1 to 10, and at the start of the trial, everybody's a 10. And then you do the trial, and everybody actually got better, there's still a 10 on this scale, because 10's the highest you can go. That's a ceiling effect. That's a problem. You're not going to have a successful trial if you do that. On the other side of things, there's the floor effect. So for an example here, we could think about marathons, just to have a real world example. So there's 10 of us who might be on this call who we're going to measure whether we can run a marathon in one day. Eric Marsh is on the call, and I know that he can run a marathon in one day. So Eric gets a 1 for running a marathon, and the rest of us can't run a marathon in one day. So we all get 0s. Then we train for 6 months really hard, and we're trying to get better. And maybe a lot of us actually improve and we can run further, but we still can't run a marathon. So we still get a 0. Eric still can, so he still gets a 1. So we don't see any change in this. And this can be the effect of a floor. You have to have the bottom of the scale and the top of the scale fit what you're trying to measure. Discrimination. This is how far apart the intervals are. And as an example of this, if we have a scale for CDD and we ask families, can your child sit, stand, or walk? And this is similar actually to the scale that we've used, partly because it's data that's available, and Helen presented on this previously. We could get good data about whether someone can sit, stand, or walk. Well, okay, so maybe your child's able to sit for about 10 seconds without any help before the trial starts. And the trial actually does help them. And now they're sitting as long as they want. They can sit completely independently, but they're still not able to stand. Well, that's an important improvement. And I think this is part of what Carol Ann was talking about. This is an improvement that's meaningful and important to a family and to that patient, that on this score was a 1 and is still a 1. That's not an appropriate amount of discrimination. It's not an appropriate amount of setting of intervals to let us see important and meaningful changes. So we missed it if we did the scale that way. And Carol Ann, to answer your question, how do we get that information? I'm going to tell you a little bit about some of the things that we're trying to do and how we're actually going to be collecting input from families about it. So distribution puts all of these different elements together. Can we appropriately represent, accurately represent all patients with CDD on the same measure? Is it spread out across that measure? Is everybody at the top or everybody at the bottom? Does it have the right intervals to see changes that we could see realistically in a trial? Those are all things that we have to try to take into account when we're thinking about developing outcome measures. So in order to do that, we have to have some infrastructure. And you've heard a lot about various registries and databases today, and they're all important. And you're going to hear about another one. So the International CDKL5 Clinical Research Network It's a mouthful. I'm sorry. It's my fault. I made up the name. And we call it the ICCRN, which isn't actually that much easier. But it's our attempt at trying to collaborate as well as we can to make it so that any data that you provide can be put to the best use, but with appropriate protections. So all of the centers of excellence in the US, all 8 sites, are part of this, as well as Telethon Kids with Helen and Jenny. And we really were trying to combine the great work that Helen and Jenny are doing, directly interacting with families, getting parent report measures and surveys, and combine that with what we collect for clinical data in-person visits and what a clinician can assess that maybe a parent would have a harder time assessing. And by combining those, we actually have a lot more power to be able to figure out what's going on and solve some of these critical questions. So what are the goals of the ICCRN? We want to continue to collect natural history data. There's been a lot of progress made on this over the last several years, both from the Rett Natural History Study as well as from the ICDD. But we want to continue to do it, and we want to bring all the appropriate data to bear. We also want to say which treatments that are currently available work, what works best, how do we try to figure out what is the current standard of care or what it should be. And then we want to get ready for disease-modifying clinical trials, and that's that clinical trial readiness piece that we discussed. So in order to be clinically trial ready, we need to generate and validate a suite of outcome measures and biomarkers necessary to comprehensively assess whether a drug is working. In other words, if we're only looking at one symptom, what if that's the symptom that doesn't get better? We need to look at a whole slew of symptoms. Maybe we can't get all of them perfectly, but we need to get as many as we can, particularly the really critical ones, the ones that you as families are telling us are the most important. And then we actually want to run a mock trial, and we want to see how these measures perform. We want to make sure that they're actually showing changes over time when they're supposed to, and that they're stable when they shouldn't be changing. And those are actually important things that are that have to be done. So for the ICCRN, this is all in conjunction with Helen and Jenny, as well as all the other centers of excellence. We've proposed to develop these outcome measures. Gross motor, fine motor, these are both video scales. This is what Helen showed a snippet of in her talk, is the process of us trying to make sure that video assessments of gross motor can be done appropriately. We have a number of measures that you guys get to fill out for us. You tell us how things are going. Those are in terms of communication, the QI disability quality of life inventory, and half of the CDKL5 clinical severity assessment. So that actually has 2 parts to it. There's a parent report portion, and there's a physician completed portion. And those are 2 separate measures now because they're filled out by different people. And we're in the process of making both of those better than what we originally had set them out. Now we recognize that these measures don't capture everything about CDKL5, but it's a lot. And we are really trying to make sure we're capturing the most important parts. We also know that there's some other measures that may be able to be used, like sleep measures and things like that, that we don't have to reinvent. And we're trying to use those alongside these to help validate these measures as well. So how do you make an outcome measure? Well, there is a specific tried and true process for how to do this. This has been done numerous times over the last 30, 40 years. And we're not reinventing the wheel here. We're doing what works. Think aloud. This is really a process of doing an interview with the appropriate person. So we're doing this for various measures at this point in time. Some of them Helen and Jenny have already done. This is a process by which, through a standardized interview format, we show you what these measures are, we show you what the questions are like, and you guys provide us feedback about whether or not that question makes sense, whether or not it captures your child appropriately, and gives us some critical feedback to make sure that the measure that we have at the end actually works. That's a process that's going on now. And you may actually get some phone calls from us about that, or be invited to participate. We then go through a process of actually testing it on a wider scale and going through some refinement processes. The bigger process looks like this. It goes through that interview stage. We then collect a bunch of the measure across as many patients as we can. We look at how well it performs in many of those various ways that I talked about before. Do we have a good distribution? Is it all clumped up in one area? What do we need to do to make sure that we're going to see change appropriately with that measure? And then we need to make sure that if we do that measure multiple times and it shouldn't change, that it doesn't change, right? That's inter-rater reliability. These are all various processes of validity testing that we would go through. And then we want to actually test that over time to see how that captures changes in patients as they get older. Clinical trials are more than just measures. Their measures are a critical part of it, but there's also an entire protocol on how often you collect those measures. How do you make sure that that measure is being implemented the same way at various sites, and that things are reproducible? And that affects that validity. And we have processes that we've laid out that we're going to go through in order to try to make sure that all of our different outcomes are actually being collected in an appropriate way at reasonable intervals. And this is really going to inform what would a clinical trial look like if we were going to have the best chance of being successful. So this type of work requires some pretty serious infrastructure. It requires a lot of collaboration. And we have gone through a lot in the last year and a half to try to put in place all the things that we need to make that type of infrastructure work. We need IRB. This is really a critical part because it actually protects patients, and we don't want to gloss over that. We want you all to know what's happening with your data, what are the protections on that data, what won't happen with your data, and what you have a right to do as part of that. And that's the process of making sure there's a good IRB in place and that you guys provide consent that tells you all of those things. We need legal agreements about sharing of data, and we have to actually set up the databases and the protocols to collect this data. This is what the International CDKL5 Clinical Research Network looks like. This is our diagram of it. So we have all the centers of excellence on the left: Boston, CHOP, Cleveland, NYU, WashU, Baylor, and UCLA, as well as us in Colorado, Children's Hospital Colorado. We're the data coordinating center where all the data sort of flows into. And we have the database there that's centralized. There's also data that flows in from ICDD and from our database at Children's Hospital Colorado to the ICDD and Telethon Kids database. And that allows us to cross-share between our two databases so that we can maximize the efficiency with which we collect data from families. And those IRBs, I'm actually quite happy to say, are actually in place between Telethon and us to make sure that we can share this data in ways that families are informed about it and have control over what's shared. This provides a really nice balance, as I said, between the expertise that Helen and Jenny bring to the table and the clinicians that are seeing patients every day. Ultimately, this moves towards a database called ENDAR, which is an NIH-supported database that would allow for this data to be kept in perpetuity without any identifiers, only de-identified data, so that that data can continually be useful for other researchers beyond us, as well as industry and any number of other partners. And that's really what we're trying to do to meet our goals. For this clinical trial readiness piece, we also have other important partners that we want to make sure are involved in this process. And that includes the companies that will actually run clinical trials. So you can see in the orange circle a number of the companies that either have trials or may have trials in the future that would be important to get their input about what an outcome measure might look like or how a trial might be designed. So they're going to be involved in that process as we move towards this clinical trial readiness concept. So this is a bit of a complicated slide about what that data flow kind of looks like. But again, the point here is that we have different types of data and those will be collected differently. So we have clinical data that will happen at the hospital. Some of that will happen on a research basis. Some of those things will happen as part of your clinical care. We're trying not to reinvent the wheel anytime we don't have to on that. We have parent report outcome measures. That's what PROM stand for. It's not the prom you go to when you're in high school. So that's communication, quality of life, the parent part of the CCSA, and some other questionnaires that again help us to correlate how one aspect or domain like sleep may interact with some of these other outcome measures. And then there's the motor scales where we're going to be trying to develop ways of getting these at-home videos that assess gross and fine motor that Helen and Jenny are really taking the lead on. So that data then flows through to a database where you can submit these things online to our Children's Hospital Colorado database, and we'll have an interaction with the telethon database to be able to score those videos and cross-check with their database some of this data. And then finally, it goes on to NDAR. The important thing here is that if you consent to be part of this process and to enroll at any of the centers of excellence for the ICCRN data, you're going to be consented about where your data goes and how this flows. So you don't consent to do anything or share your data with anybody without knowing about it. So where are we on the measures that we proposed? All these different things I mentioned before, we are in this phase for most of them. Some of them we've actually gone to some larger collection of data sets like the QI disability. For the CCSA specifically, we're in the process of starting interviews with families. So we're collecting your feedback about how the CCSA works, whether it's understandable, whether you can complete it on your own, whether it's too long, too short, whether it gets those intervals correct so that meaningful changes are actually represented in it, to Carol Ann's point. So that's going to be our process for collecting that. We're going to be doing that over the next several months. And again, you might actually get us reaching out to you. If you're interested, please feel free to reach out to us. If you don't have my email, we can get it from any of our IFCR folks. They know how to get a hold of me. And we can also put some information on the website about how this would work. And after we get through that interview stage, then we test it on a broader group of patients. For the provider part, we also have to go through that same process. So we're getting all of the centers of excellence providers to complete interviews with Helen and Jenny, where they're going to assess how well we do at filling out these forms the same way, whether we're all thinking about the forms similarly or not. And then we're going to move towards larger collection. There was a question ahead of time about what the CCSA will be used for, the severity assessment. And I think this is a really important point. The goal of the severity assessment, if we do it right, and we actually have the right distribution and can see change in a patient, is to make it so that we can assess whether or not there's improvement in a patient that's meaningful, regardless of the severity of that patient. So it's not to determine which patients would qualify for a clinical trial. That is not our intention in the design of this tool. It's so that any patient at any severity could be appropriately tracked for improvement. And I think that's a really important point. I also want to be clear that a clinical trial for a gene therapy or anything like that in the future will not be something that I design. I will not be the person who's running it. That's the sponsor, that's the companies that are involved in that. I think that oftentimes we are consulted for that. We may have input on it, and we're going to provide that input best we can. But ultimately, the details of how a clinical trial works is quite complicated to come up with, and companies have to try to make sure that they can do it in a ethically responsible way that's going to make sure that that trial is successful if it should be. And so that's something that's decided by the companies themselves. So what do we need from all of you? participation. And I know this has been the theme already of the last several hours, and we've talked about all the different things we want you to sign up for. Participate in interviews that I just mentioned. Fill out the surveys over time and these various outcome measures. We may ask you from time to time to keep a diary about certain symptoms. And we may ask you to participate in biomarker testing or collection. And again, what's involved with that, you might get a little bit more flavor from with Dr. Marsh in a minute. I recognize that this is a lot. I think one of the things we've tried to be very conscientious about, in addition to everything that Dan Lavery said about coordinating our data and trying to make sure that we're reducing burden on families, is that not every family has to participate in every part of this. We want to collect the minimum amount we need to be able to move to the next phase in a way that's going to actually result in success, right? So we don't have to do interviews with all 300 patients that are in the ICDD database in order to move to the next phase in an outcome measure. We might have to do interviews with 15 or 20 patients. It sort of depends on what the goal is. But participate where you can. And we understand that it's a lot we're asking, and we're trying to be sensitive to that. So how are we trying to streamline? One, we only collect what we need as often as we need it. We're trying to coordinate so that clinic visits and other studies happen together. That's something that we're going to have to try to coordinate locally at sites, but we're also trying to coordinate that with clinic visits and with outcome measures that we're prompting you to complete as part of the ICCRN data with Helen and Jenny. So for example, if we do a survey about how things are going and we need an update on that, it makes sense for us to do that prior to a visit that you have upcoming for clinical purposes anyway, where the answers you give to that survey could actually be used to inform what do we talk about during that clinic visit. We're trying to coordinate those things the best we can. And we may not get it perfect. Give us feedback. We'll keep trying to make that better and do it the best we can. Many of our measures can be completed at home at your convenience. So we may ask for it to be completed within a certain time window, but we don't have to do it at exactly one particular time. So we're trying to create some flexibility there. There's also those options that were mentioned by Dan. You have to be able to stop, come back, complete it again later. And we're trying to build all that into the functionality of how we do this. And then we're responsibly trying to share data where we can across these different databases and different groups so that you don't have to fill out the same questions multiple times. We have to do that in a way that's consistent with all the regulatory processes that were described throughout the morning. And we have to do that with your permission. But we're doing our best on the back end to make it possible to do that should you want us to. And then finally, we have to try to keep you up to date about what's going on and why it's important. That's why talks like this one hopefully are also achieving, is that you understand why we need to do this, why it matters, and how you can help. So what's some of this look like in the big picture of that infrastructure? How's that data sharing look? Well, you can see the circle in the middle. This is a busy slide, I know. The ICCRN is a combination of our centers of excellence in this ICDD database. The ICDD actually has a strong line over to the CDKL5 registry. And then historically and still ongoing, we're collecting data on the natural history study that's supported by the NIH. And we're working on ways that we're going to be able to integrate that data into what we're also trying to do in the ICCRN. Not every aspect of this data flow is currently up and running, but this is our goal. This is what we're trying to work towards to be able to make it so that we can do things as efficiently as possible. And with that, we thought it would be useful, both from the Lulu and ODC side of things, the ICCRN, ICDD side of things, to give a little bit of an overview of just what are all these different things? What are all the different types of observational studies that are going on? And how are they complementary, not competitive? So you've heard about Connect CDKL5, and that's really a way of IFCR knowing who do we have with CDKL5, where in the world, how can we stay in touch with them and integrate them into the community, should they want to be. That gets you research updates. It also gets some basic information to be able to contact you. There's the NIH Natural History Study. Not all of the sites are COEs, and not all the COEs are sites for the natural history study, but there's a lot of overlap. And we're taking a lot of the lessons learned from the natural history study and implementing them towards the ICCRN goals as well. They're trying to determine the natural history of Rett and Rett-related disorders, which includes CDKL5. They're developing tissue and functional biomarkers, and they're developing outcome measures. And again, many of those clinicians are the same clinicians that are in the ICCRN, and we're carrying over those lessons. The ICCRN, including the ICDD and the COEs, which I've described the goals of throughout this talk, the CDKL5 registry from Lulu and the ODC, really working on trying to get some essential data from as many patients as possible, linking that in where possible with the ICDD, which can also be a way that you can find out where are the centers of excellence if you've never been seen at a center of excellence before, how could you get into a center of excellence if that's something you're interested in. And if you're not, and you only fill out data through the ICDD, that's fine as well. Or if you don't live in the US and can't make it to the US, that's fine. You can fill out registry information and information through ICDD that will still be useful in terms of all of our combined goals of getting ready for clinical trials and knowing more about all of the impacts across life of CDD. The final thing that I want to mention is that Lulu Foundation with ODC and a number of industry partners are working on something called an endpoint enabling study. And just so that you're not surprised when you hear more about this in a couple of months, this is a study where you're doing a clinical trial readiness study like we've described, but instead of generating new measures for CDKL5, you're using existing measures, things like the Vineland and seizure counters and things like that have been used in trials previously. This is actually an important study that helps us to answer which things, as we move forward with the goals of the ICCRN, which things do we not need to reinvent? They actually work pretty well. We know not all of them are going to work perfectly, but how well do they work and which ones work better than other ones? And this is really the goal of an endpoint enabling study, is to be able to get up and running with what outcome measures do we have now that work reasonably well, because it's going to take some time for us to validate the newly generated measures. And so that's on its way as well. And you'll hear more about that from Lulu, the ODC, and some of the industry partners who may be involved with that. And again, we're trying to coordinate so that we can learn maximally along with them about how to collect that data and how it would interplay with some of the other data that we're collecting. And with that, I give a special thank you to all of you, the parents and patients that are participating in research and that help us to move things forward. You guys are really the reason that we do all of this. I want to thank all of the centers of excellence, and these are all the docs across all of our centers, as well as Helen and Jenny, Lulu Foundation, and IFCR's ongoing support for the ICCRN. And I think with that, I am turning it over now to Eric Marsh to give you guys the brief 101 version of biomarkers and why they matter. And then he and I can answer some additional questions together after he's done. I will try to stop sharing.
We're going to wait. Amanda's loading up Eric's slides. So while we're waiting on that, I'm going to give you guys some questions that came in. There they are.
Okay.
As long as we know you have control, Eric, we'll hold questions.
Eric, do you see a mouse in the bottom left of your corner?
Yep,
bottom
I do.
left-hand corner?
I do.
Okay.
I tapped it. That should be good. You're gonna put on. Okay. Hello, everyone. My name is Eric Marsh, and and I am a neurologist at the Children's Hospital of Philadelphia, and now also clinical director of the Penn Orphan Disease Center. And after that wonderful talk by Scott that laid out Dr. Demarest, that laid out the landscape of all the clinical and natural history type of and outcome measure studies that are being done for individuals with CDKL5, with CDD, I'm going to do a 101 class for biomarkers, as this is an important component of it. And I get the mouse, but how do I move this forward? Oh, hold on, can I go forward? There we go, thank you. There's my little mouse thing. Okay, so just disclosures that I do serve as a local PI of a bunch of things, but now to get to the substance of the matter. So what is a biomarker? So this is from Google, which is where, oops, we'll go back here. No, go back one. Yeah. Oh, we could just stay there. So this, the first slide was the definition of, oops, can you go back? Amanda, if you're controlling this, can you go back? So a biomarker is any biological characteristic that can be objectively measured and tested as an indicator of both normal and abnormal medical processes or a response to a therapeutic intervention. Biomarkers aren't necessarily an endpoint of a study, as in what a clinical trial would look for, but can be used in parallel with one of those endpoints. So, for example, if you were doing a trial with an anti-epileptic drug, a decrease in the number of seizures would be the clinical endpoint. But if you had a marker in the blood that would tell you that you were going to have a decrease in the number of seizures, you could use that blood-based biomarker instead of counting seizures. Amanda, can you go to the next one? Because I don't seem to be controlling this very well. So,
Eric, it wants you to accept, if you want to control the screen, if you would like to control it up towards the top, you should be able to take control.
Okay, I got it. It says I can control it, but it doesn't.
Okay, I'll be happy to, I'll help.
All right, thank you.
Okay.
All right, so there are many different types of biomarkers. Biomarkers can be used to track the progression of disease. And the best example of this, if anyone knows anyone who has diabetes, oops, can you go back one? No, so well, that's the right slide. So there are different types of, where are we here? Okay, let's say there. There are types of biomarkers. You can have biomarkers that can measure the progression of disease. So if you know anyone who has diabetes, they, every time they go to the doctor, will get measured a hemoglobin A1C. And it turns out that depending on the level of your hemoglobin A1C, you can predict how good of control someone has of their diabetes. If the A1C goes high, the diabetes is under poor control, and that puts them at risk for things like peripheral neuropathy or getting pain in their legs or heart disease or strokes. You can have drug activity markers. These are markers that have you know that a drug is actually working. And a good example for what all of you might be aware is if your son or daughter is on a drug like clobazam or Valium or Ativan, which are called benzodiazepines, if they get an EEG, there might be some very fast activity on that EEG. And that's a sign that it's a marker that the drug is actually engaging its target. There can be surrogate markers. So they can tell if the endpoint has been reached. And an example of this is tumor recurrence in someone who has cancer for an MRI. So you can look at the MRI and you're not seeing the tumor, and you don't know how the outcome is going to be. But if you see the tumor spread or grow, you know that that's going to be potentially a bad outcome. And then you can have predictive markers. And this is going to be whether a drug or treatment will potentially work for a given disease in advance of actually starting that treatment. And the best example of this is there are certain genetic changes that people can find in tumors that will tell you if a certain chemotherapeutic agent will cause a response in that patient for that chemotherapeutic agent. Another example is there are markers for response to the blood thinner Coumadin. If you have a particular marker, you're going to not respond to Coumadin, or you'll need a really high dose of Coumadin. So there's some very good markers that people have come up with to look for the progression of disease, whether a drug's working, whether you can follow the course of the disease. And that's the type of thing that we're trying to look for CDD. Next slide. And so really, there's two broad classes of disease biomarkers that one can have. There are what we call functional biomarkers, and this is for neurologic disease specifically. There are functional biomarkers, and these are things that are measuring the activity of the brain. And as you all know, the brain's activities are walking and talking and thinking and sleeping and all the things that we do. Those are what the brain does. And so can we measure those things? And there's a lot of different ways to measure the things. The easiest ways to measure these things in people are the EEG or the functional imaging. And then there are other functional measures such as transcranial magnetic stimulation or magnetoencephalography, but those are all functional biomarkers. On the other hand, these are called fluid biomarkers. These are things that they can get from blood or urine or stool or cerebral spinal fluid. And from those fluids, you can extract things like DNA and RNA and cells, and you can test for multiple different things within these fluids to see if they can act as biomarkers, or either that tell someone is going to respond to a drug or that someone is responding to a drug. Next slide. So this is an example of a functional biomarker in CDKL5 mice. So this was a study done by Judy Wang in Joe Zhao's lab back in 2012, right when they first made the first CDKL5 knockout mouse. And this is an auditory evoked potential. And this is where they played a sound to the mouse. And in the upper left-hand corner there, you can see the response of the CDKL5 mouse, which is the purple line, and then the wild type mouse, which is the black line. And you can see there's a clear difference. And they were able to show that there was a significant difference in a couple of the amplitudes of the response. And so that they could separate out which was the knockout mouse, which was the CDKL5 mouse, and which was the wild type or control mouse by just looking at this marker. So this was a marker of disease in these animals. And in parallel to that, in the NIH-funded natural history study that Dr. Demarest mentioned, we've been measuring this in children with CDKL5. So next slide. And what we see in this slide is a visual evoked potential. So this is where the child gets EEG electrodes put on his head, and then a visual stimulus, which is a checkerboard, and the black and white squares switch spaces so that there's a change in contrast. And you can follow what the brain does. And so on the top, these are average waveforms for the Rett subjects, it's 38. From the CDKL5 subjects, it's 11. And for the control subjects, it's 22. And down below are the individual brain responses. And you can see that the RET waveform looks very different than the CDKL5 waveform, which also looks different than the control waveform. And we're in the process of finishing up this first pass analysis of the CDKL5 data. We have over 20 CDKL5 patients that we've recorded, and we are looking to see whether any components of this waveform can tell us something about the severity of the disease, or the age of the patient, or any other clinical markers. Next slide. So as Dr. Demarest was mentioning for kind of why we do this, this helps CDD because biomarkers can make clinical trials easier and more accurate. We can potentially develop surrogate endpoints that would allow us to not just have to use the clinical outcome measures, but use those clinical outcome measures that Scott very nicely described in the process of developing them, along with these surrogate measures, where together you get a much better assessment of whether a drug's going to work and how well it's going to work. One other use is that if you could do these markers early, you could potentially predict outcomes, and that to help in families inform decisions and make care choices. These are the type of things that would really be needed for a gene modification or gene therapy trials that, we don't know if you restore CDKL5 back in an individual, how long will it take, if it happens at all, but how long will it take for the child to start to regain skills? Will it take days, weeks, months, years? And if it takes a longer time, having a biomarker, whether it's a functional biomarker or a blood-based biomarker, will really aid in the companies and the physicians and the families in understanding what's happening in these trials. And so families can help by participating in the natural history and outcome measure studies that Scott had already talked about, but also these biomarker measures that are going to be done in parallel, not necessarily at all the sites, but in some of the sites, parallel with these other clinical outcome measure development. Next slide. So again, as Dr. Demarest said, just most importantly, I want to thank everyone who's participated, the patients and their families, the funding for this, and much of the analysis is being done at the Children's Hospital of Philadelphia with my colleagues. And then this data, that the one slide of data that I showed you was from the NIH Natural History Study team, so the members of that team too. So thank you. I think with this, Dr. Demarest and I will take some questions. And just for, I look like I'm a ghost in this, I just didn't see that. And as for Dr. Demarest's point of ceiling effects and floor effects, everyone can run a marathon if they just work at it. So, all right, questions? Oh, Karen, you have a visitor in the background.
Many thanks to both of you, and Heidi and Amanda do a great job of monitoring for those questions, so I'm sure they're about to give us some.
We have some on our YouTube feed. Carol Ann was very excited about some of the measures that Dr. Demarest introduced. She said the Rare Disease Collaborative Network in the UK are just about to enter natural history, and she knows Dr. Sam Amin would be interested, Dr. Demarest. We had carried over the question she had asked before about capturing the children with small changes, and she had said that, and how to speak with the industry partners about these measures. She mentions that Amber has been in a trial, and they've been using parental observation, education, and physiotherapy input to determine the improvements. Are there available questionnaires? The available ones that they're using won't capture, although she is test driving severity scale. She believes it is important for comments and observational evidence from external sources, meaning the education and therapists.
Yeah, so there was a lot there. We agree that getting input from some of the additional types of providers will be helpful. That's part of the types of activity that we will also have as part of our development of these outcome measures and development of clinical trial readiness. Some of the measures are actually performed by a physical therapist or speech therapist that we are doing in conjunction with some of the measures that we talked about. So they're going to be integral to that validation process. We completely agree there. We know that many of the existing measures aren't perfect, and that's part of why we're trying to do all of this. There is the CDKL5 severity assessment, which was published almost a year ago, that is available. We will say we already know that it's not perfect, and we are in the process of trying to make that better. And so we will continue to make that available as we get to new versions of it, so that can be used for any of these types of activities. Did I answer all of the questions that were covered there?
She has one more follow-up that fits in nice right here. She wants to know if you require concurrent parental observation in respect of the biomarkers to determine efficacy. I think you can speak to that with the tool you just talked about.
Concurrent parent observations. So in other words, do we need to get both parents' observations about some of these things? Yeah, I think that's a really interesting question. Part of what we try to do with any type of outcome measure is make sure that no matter who fills it out, we get a reliable answer. That's not an easy thing to do, actually, but that's why getting that input from families in the interview process, so that we know that people understand what we're trying to ask and can answer appropriately, is really critical. I think it's a very interesting question about whether or not we should actually look at some rate of reliability between parents. It's not something we actually had outlined previously in our plans, but I think Helen and Jenny and Tim and Eric and I should chat about that. It's a really interesting question.
Would another solution to that be to try to make sure it was the same parent that filled it out?
So in many clinical trials, that is the process, that you would ask that the same parent is filling out any particular measure each time. But I also recognize that there may be challenges associated with that, particularly if you're thinking about a gene therapy trial that may take place over several years. That may not always be possible. And so it's reasonable for us to try to do some basic assessments about how well parents align when they're filling out some of these measures.
That's a very good point. Very good question.
Can you speak to participation in the natural history and filling out some of these different scales? Some of them require point of contact. Well, are there ways for people who are unable to travel to participate in any of the given research studies?
Eric, do you want to take the NHS activities? And I'll just say before you do that we are trying to make it so that anything that doesn't have to be done in person isn't done in person for the ICCRN activities as we go forward. But I'm going to let Eric speak to what's being done for the NHS.
Yeah, so for the natural history study, originally it was designed for everything to be done in person. A few years ago, we added an amendment to the protocol to allow some remote data collection for individuals who we didn't think it was either too much effort or potentially it just could not travel, especially for the places that have to travel more further distances, so that there is some remote data collection. Some of the main scales that we use for the natural history study do require physician observation, but now as we're all learning, and as COVID has made us rapidly adjust to using telemedicine for our ability to observe, we can do a lot of it via telemedicine. So I think there, will be a continued shift to doing things more remotely. But the current iteration of the natural history study, there is now an allowance for remote data collection, but there wasn't originally, in which if we were to redo it, we would have probably done remote data collection from the get-go.
I don't see any questions, additional ones anyway, on the, well, yes. The question related to the biomarkers, what is being clarified, it says that they were referring to correlation between biomarkers and what parents report. Are they congruent? But the person who's answering the question says they're sure you understand what they're talking about, Scott.
Yes. So it's a good question. So, one of the things about the different biomarkers, oh, my lighting's off a little bit here. One of the issues about the different biomarkers is that they don't necessarily track one-to-one with, say, a parent-reported measure. They can be tangential to it or additive to it, so that there isn't necessarily a direct correlation. For the visual evoked and auditory evoked potentials in the Rett natural history study, we've finished the first analysis of the Rett subgroup. And in that, we do see that there is a very strong correlation between the Rett motor behavioral assessment and the Rett clinical severity scale, with amplitudes and latencies of both the visual and auditory evoked potentials. So in that case, there is good concordance between what the parents are reporting, or at least what physicians are reporting, and the functional biomarker. But that doesn't have to be. And that's, one of the things that we're still learning about is, can these markers tell us something that can be additive or predictive of, or even better than parent-reported outcome measures, right? That they could potentially tell you, this person's going to have a great response, and it's not till after the person has a great response that you know. So we're still learning about how to best use these markers, but in some cases they are congruent with what the parents report. And that's, at this point, we're often using the parent-reported outcome or the physician-reported outcome as our comparator. So we're trying to design outcome measures that match to that, but that doesn't necessarily have to be the case, if that makes sense.
And I would just add to that, that it may be that it correlates with one particular aspect or symptom of that patient, but not with overall severity or something more global. And that's what's really critical about the kinds of studies that we're trying to do, is us clarifying, what does that biomarker actually tell us? What does it correlate with? How are we going to best use this in a clinical trial if we were to do that?
And that's actually an excellent point, Scott, because for the Rett study, what we do see for the MBA, the Rett Motor Behavioral Assessment, is that the evoked potentials correlate really tightly to the social subscales and less tightly to just the gross motor subscales, which actually intuitively makes sense, because we're measuring higher order function, which is what social and language processing is as compared to gross motor. So it makes some sense, but there's a good example where it does correlate to the total, but it correlates best to a subscale of outcomes.
Okay, we have one more question. This parent wants to know, if they change the center of excellence they go to, does their information for natural history follow them?
Yeah, so again, the natural history study might be a little bit different than the ICCRN, because of the way we're designing the centralized database through Colorado for the ICCRN. Your data will follow. So if you switch what center of excellence you go to, we'll be able to see the data that you provided at the prior center of excellence and data that you provide at a new center of excellence, because life happens and people move, and we need to make this work for you anyway.
Yeah, and the natural history, the NIH Fund study did also, it tracked people. And so if someone moved, the data went with them, and the new site got access to all the data so that they would know what was there. And centrally, the same patient got tracked so that there was no reduplication of patients in the database.
Okay. I don't see any more questions. Heidi, do you?
I don't, and I think we have another session, exciting one I've been looking forward to that we need to get started. So I think we better thank Dr. Demarest and Dr. Marsh and get headed over to the next session.
Yeah, guys, give us about 5 to 10 minutes and check back at the YouTube channel for the new feed, but we will be up and going as soon as possible. We're going over there right now.
Thank you.
everyone.
Thank you all.